Hello, everyone. I am Yifan Wang. I am a research associate at Mayo Clinic. First of all, thank you for the opportunity to present our exciting findings at the Frontiers in Neurosciences conference. Today, I would like to go through our findings of interneuron loss and microglia activation by transcriptome analysis in the basal ganglia of Tourette disorder. First of all, I would like to go through some basics of Tourette syndrome or Tourette disorder, which will be abbreviated as TS in this talk. TS is a common disorder affecting as many as 1 in 150 children. The main symptoms of TS includes motor and voter tics, which are usually out of context and in a repetitive fashion. TS has one of the highest familiar recurrence rates among complex neuropsychiatric disorders, and for now, no recurrent genetic variant or biomarker has emerged as a causative factor. And from previous studies that we know that pattern of motor programs are encoded in the basal ganglia region of the brain. And basal ganglia were also shown to be activated during the tics of TS patients. Previous neuroimaging data also suggests that caudate and putamen regions, together referred to as striatum, are smaller in TS patients' brains. Previous studies using immunostaining and bulk RNA-seq data from the Vaccarino lab also showed that there is a decrease in the NOS1 plus interneurons, both in the caudate and the putamen region, as shown in the upper panel here, and an increase of CD45 PT-PRC plus immune cells in the caudate region, as shown at the bottom panel here. And from the bulk RNA sequencing data, they have also observed the top scoring downregulated module in TS was enriched in striatal interneuron transcripts, and the top scoring upregulated module was enriched in the immune-related genes in the TS brains. So with all the previous studies, we could assume that the etiology of TS can lie in aberrant function of specific cell types, for example, interneurons and microglia observed in using the bulk methods. However, in this study, we're wondering whether cell type specific information can explain better the molecular mechanism of TS. So I would like to start our findings with the experimental design and workflow. So we were able to collect in total 6 pairs of striatum regions from the brains with age-matched Tourette patients and normal controls. With 3 pairs of striatum, we call it as standalone single-cell RNA-seq and standalone single-cell attack-seq, where we send part of the tissues for single-cell RNA-seq and part of the tissue for single-cell attack-seq data. And at the time when we generated the sequencing data, there was also multi-omic single nuclei sequencing coming out. So for multi-omic sequencing, we send the same piece of tissue for both single-cell RNA-seq and single-cell attack-seq data. That means the RNA-seq and attack-seq data came from the exact same single cell. So by combining these these 6 pairs of single-cell RNA-seq data, we were able to take a look at the cell type composition change in the TS brains, as well as the genes which are differentially expressed in the TS brains. And with the single-cell attack-seq data combining all 6 pairs, we were able first, as well, we could check the cell type composition. And then with the single-cell attack-seq data, we were also able to check the differentially accessible peaks. And combining single-cell RNA-seq and single-cell attack-seq together, we were able to identify the DAPDG association within the TS brains compared to controls. Then I would like to briefly go through the single-cell RNA-seq technique as well as how clustering and cell type annotation works. So for 10x genomics single-cell RNA-seq data, it will suspend each single cell in a in a float here, and then they will collect. So within each single cell, reverse transcription would happen, and then the cDNAs will be amplified for downstream sequencing. And within each bead, a unique barcode will be attached to for a specific to all the reads within a specific cell. So when they were sequenced together, we'll be able to assign each read back to a specific cell by the unique barcode. And then for all the reads, we will map it back to the human genome. So with that, we'll be able to assign it to a specific gene. So with that, we'll be able to get this gene expression count table as shown here. For each row, it shows the gene expression level of all the genes within the specific cell. So now we have this count for all the genes for every single cell. We can run some clustering algorithm to cluster these single cells based on the similarity of the genetic profile of the expression profile here. So as you could see in this toy example, the cells are clustered into 3 distinct clusters. And then using the genetic markers that are expressed in each specific cluster, we'll be able to assign the cell types into each cluster. So with that, when we applied the clustering and cell type annotation to our data, we are able to see in this UMAP, we saw the separation of neuronal cells, medium spiny neuron, and interneurons from the glial cells, oligodendrocytes and astrocytes, as well as we're also able to see microglia here. And then in the middle, we're able to see the genetic markers for each specific cell type, as well as from our data, we're actually able to see a clear separation of DRD1 and DRD2 medium spiny neurons in our single-cell RNA-seq data. And on the right, I am showing a panel of how genetic markers are enriched in each cell type. The larger the dot size is, the higher average expression of specific genes are within a specific cell type. For example, we could see the higher expression of CD74 and RGS10 in microglia here compared to all the other cell types. With the cell type annotation, we now are able to take a look at the cell type composition, where we observed a reduced interneuron counts in the TS brains. So in this left panel here, I am showing the normalized difference between the TS and normal controls for each cell type as marked in the x-axis, and we were able to observe a significant interneuron loss in all of our TS brains compared to the paired controls. While for single-cell RNA, single-cell ataxic data, which I'll be talking into more technical details later, we did not observe a statistical significantly interneuron loss in the TS brains compared to controls. However, we did observe the same trend in the interneurons. And unfortunately, with the five pairs of data available for comparing cell type composition in microglia, we did not observe a statistical significance. However, we are able to see that for 4 pairs out of 5 pairs of brains that we have, we do see higher microglia, a higher number of microglia cells in the TS brains compared to control brains, which is both of them were concordant with the observations from the immunocytochemical analysis published previously. So for further analysis, we looked at the DEGs that differentially express genes in each different cell types. So first of all, we were able to identified 300 to 600 DEGs in each cell type. And within these DEGs, first we observed an upregulated immune response related signature in the microglia, as shown in the red here. As we could see, we are seeing an enrichment of these pathways in the upregulated genes of microglia. And then further, we observed a downregulated DG signatures enriched in the plasma membrane and postsynaptic membrane pathway for interneurons. Lastly, we also observed a downregulated mitochondrial function-related signature for medium spiny neurons. These observations were concordant with the top signatures associated with TS in the previous GWAS studies. So with the three different enriched signatures we observed in medium spiny neuron, in interneuron, and microglia, we would like one question that we wanted to ask is that was there any correlation between these signatures? So we took the two major pathways. One is a medium spiny neuron mitochondrial related pathway. The other is a microglia immune-related pathway. We calculated an enrichment score, which reflects how overrepresented a gene set, which is the genes within specific signatures, is at the top of a ranked list, which is all the differentially expressed genes in this specific cell type. So we calculated the correlation between the ES scores of microglia and medium spiny neuron, and we observed a statistically significant correlation between these two pathways. This actually suggests us that there might be a cause-effect correlation. There might be a cause-effect relationship between activation of the immune system in the microglia and the compromised oxidative metabolism in the mitochondria of medium spiny neurons. So that wraps up our findings using the single-cell RNA-seq data. Then I would like to come into the attack-seq part. So first of all, I would like to briefly go through how attack-seq works and what the data looks like and what it actually shows to us. So the attack-seq TN5 transposase would cut the open chromatin regions and then amplify them for sequencing. That says what we will be able to sequence using the attack-seq technology will be the open chromatin regions of the genome. And with all the amplified sequence, we'll be able to call these peaks, as you could see as green here. And these peaks represent the open chromatin regions, where biologically we think these are most likely to be promoters and enhancers in specific tissues that were sequenced. And with single-cell attack-seq, since here we will only be able to get reads that map to the gene body as well as the upstream region of a gene, we will aggregate all the reads which map to the gene body as well as the reads of 2,000 base pairs upstream of a gene, and use this count as the gene accessibility count for each gene within one single cell. So similarly to the single-cell RNA-seq, we'll be able to use this gene accessibility count table to do the clustering. So the cells with similar gene accessibility profile are going to cluster closer to each other. And with this clustering, together with the cell types which we were able to assign to each single cell, we'll compare the counts from RNA-seq and the counts from single-cell attack-seq. And then we'll be able to do what we call as label transfer to be able to annotate the cell type clusters in the single-cell attack-seq data. So with that, here is how our single-cell attack-seq clusters look like. As we could also see, is that we see a clear separation between the neuronal cells and the non-neuronal cells. And we see the medium spiny neurons and the interneurons on this side. The oligodendrocyte, astrocytes, and microglia on the sides, and they separated pretty well. And then in the middle, we're also able to see the genetic markers. And on the right side, it's the same genetic markers that I was showing for the single cell RNA-seq data. We saw enrichment, for example, for the BCL11B, PPP1R1B in the medium spiny neuron, for example. So with the single cell ATAC-seq data, we were able to, in the end, identify over 300,000 peaks within different cell types. And among these, 26% of them were cell type specific. And among the 300,000 peaks, 78% of the peaks were in axonic and intronic regions of the genome, which is concordant with other published single cell ATAC-seq data. And for and when we were looking at the differentially accessible peaks comparing the TS brains and control brains, we were able to find the most number of differentially accessible regions in the TS versus control in the medium spiny neurons and oligodendrocytes, as you could see in this figure. So in total, we observed over 1,000 more accessible peaks and over 3,000 less accessible peaks in the TS brains. With the open chromatin regions that we identified in the single cell ATAC-seq data, the next question we would like to pursue is that we would like to take a look at the co-accessible single cell ATAC-seq peaks. By co-accessible, we mean how possible two peaks are present, are open in the same cell for the same cell type. So we were able to calculate this co-accessible peaks, and with that, we're trying to focus on the cis-regulatory interactions by studying these pairs of co-accessible peaks, as what I'm showing here. So if one of the pairs of the peaks will overlap with the transcription start site of a gene, and we would then treat the other end, which is co-accessible with this peak, as the distal enhancer of this peak. So by doing this, we're studying the enhancer promoter correlation using the single cell ATAC-seq data. So with that, we were able to identify 211 non-redundant DAPDG links. So instead of looking at all the co-accessible peaks here, we are focusing on the peaks which are differentially accessible in the TS brains and the genes which are differentially expressed in TS brains. With those 200 non-redundant DAPDG links, we were able to find significant enrichment of these links over total number of links in the three major cell types: medium spiny neuron, astrocytes, and oligodendrocytes. There's no significance observed in interneuron and microglia, probably due to they have very low cell fractions, which is less than 5%. Although we were not able to identify any statistical significance in the microglia, I would still like to show you this beautiful example that we found for the DAPDG links. So we found the potential enhancer region for the SLC 11A1 gene, where we saw the region here, the peak here, which is co-accessible with the transcription start site of the SLC gene, that we found in the TS brains, the potential enhancer peaks are more accessible in the TS brains compared to normal control brains. And in the same time, we also found that this gene is also upregulated in the TS brains. So we hypothesized that there might be there might be a correlation between the the more accessible regions in the TS brains and the upregulated gene when while these are co-accessible in the single nuclear ATAC-seq data. And this gene is also has multiple effects on macrophage activation and has an important role in the immune response. So in the end, I would like to summarize our study. So we were able to identify the fewer interneurons in the TS brains, and we identified different downregulated and upregulated signatures in medium spiny neurons, interneurons, and microglia. And we also significantly identified a correlation between the compromised oxidative metabolism in mitochondria of TS brains and the activation of the immune system in TS microglia. And furthermore, with the single cell attack-seq data, we were able to identify over 4,000 unique differentially accessible peaks from the TS brains, which potentially can serve as target to look for genetic variants or that are related to TS phenotype. Furthermore, we were able to identify 200 and over 200 DAPDG association links, and we were able to find significant enrichment of these non-redundant DAPDG links over all accessible peaks in 3 major cell types. With that, I would like to thank all my collaborators, specifically Liana Fashion, who collected all the samples and performed all the wet lab experiments, and my mentor, Alexei Abizov, as well as Flora Agnorino from Yale University. Thank you, and I would like to answer any question.